Yes, we have started.
Okay, great. So Prajwal, Vinod, you are also there, right? Everything is good with you?
Yes,
Can we start?
yes, yes,
Okay,
yes, we're ready. Yes.
Yeah.
great, great. Okay, so good evening, everyone, to the listeners in YouTube as well as in Facebook. Welcome to the eighth session of the Vertical 3, which we focuses mostly on practical sessions on open as well as on proprietary software. So myself, Giri, one of the coordinator for Drug Discovery Hackathon 2020. As I say every day, please, please do not share any personal details during this online meeting through the live chat. The details for the registration process and about your certificates, everything will be announced soon. So kindly patiently wait for that. And all of you kindly type your questions in the live chat. And I also humbly request you to refrain from greetings. We accept all your greetings, but we do not want to miss any questions. So kindly refrain from that so that we will focus more on to questions and we don't miss any one of. If the slides are containing any of the web articles or lectures and tutorials, please it all the copyright owns to their respective authors. As you all know, we will be focusing on these different areas of drug discovery, and today we will be focusing on a topic called importance of non-covalent interactions. And today we have two speakers. One is Dr. Prajwal, the senior scientist at Schrodinger, has received his PhD from Naipa Mohali and the recipient of prestigious with 4 years of postdoctoral research at Heidelberg University. He has been associated with many eminent scientists and has experience in diverse range of CAD projects using various types of molecular modeling techniques to understand biological processes. Holds about 30 to his credit. And today, our second speaker will be Mr. Vinod Devaraji. He is also a senior scientist at Schrodinger. He has an experience holding masters in technology, learning and deep learning applications in computer drugs. Welcome both of the Dr.
Hi. Thank you very much, Dr. Giri. Am I audible? Can anybody please confirm whether it is fine?
Yes, yes, Prajwal, you are audible.
Okay, great. So just share the PowerPoint presentation and the slideshow mode. Yeah. I hope it is visible. Is it?
Yes, you are on.
Okay, great. So, yeah. So, good evening to all of you. I hope you all have a fruitful day. So, today, first of all, thank you very much, Dr. Giri, for a nice introduction for both of us. And today we means me, Prajwal, and my colleague Vinod. So we will be here to demonstrate you the process of molecular docking, and basically our emphasis will be on what are the basic concepts of molecular docking. I hope you will not get bored because you probably have been listening it from past few days, but I will try to keep it as simple as possible. But we will emphasize more on how easy the process is using Maestro, and specifically the Schrodinger suite. So that's what is the main concept of today's presentation, and we will see how we can use Maestro for the molecular docking process and how we can prioritize few hits out of the complete ligand database. So that might be of your great interest for your coming problem statements if you are getting involved yourself in it. So okay, so before proceeding for the presentation, let me tell you, like, we have the our email IDs are displayed here, So basically it's just name @Schrodinger.com. So my name is Prajwal @Schrodinger.com, and my colleagues is Vinod @Schrodinger.com. And our mobile numbers are also pretty simple. So basically everything is same except the last two digits. Like for me it is 33, and for Vinod it is 44. So the thing is, you can always contact us regarding all queries related to computer-aided drug designing and the Schrodinger software. Please don't hesitate to contact us. So we are always available for you. So now I will proceed with the presentation. To know a little bit about basics of you can say like drug designing, or basics of something called molecular docking, or how enzyme and ligand bind. So that's very simple thing. Probably everybody has heard about it. Like it follows the lock and key model for enzyme and the inhibitor or any drug molecule. So just like the concept of lock and key, like you have lock can resembles to the biological target, and the key means your inhibitor molecule, or you can say drug molecule. So as a drug molecule fits inside the lock, means your biological molecule, which can be your protein, DNA, RNA, or anything. So any macromolecule it can have. So the thing is, as it fits inside it, it changes the function. It might be induce some conformational changes in the protein, and then in this way it inhibits the further function of that particular enzyme. So the whole idea behind is it to identify the key. So in the world there are many millions of such keys are available, but which key is of your interest? So that can be only explored until unless you know the internal structure of the lock. So lock means your protein. So if you know what is inside the lock, then you can definitely design a better key against your lock, or you can identify it from the database of keys, means the ligand database. So again, how key interact with the lock is the same way like any inhibitor or drug molecule interacts with the biological biomolecule, and there are many non-covalent or covalent type of interactions which occurs between two. Those can be your hydrogen bonding interaction, ionic interactions, or van der Waal interactions, or there are many such interactions which are present between them. So we just want to use all that information which is available with us. Somehow we want to use it with the help of software, with the help of manual exercise, or anyway, we want to use it for identifying the better key, that is your better drug molecule. So in computer-aided drug designing, the term CAD, so mostly it is divided into two types of method, like structure-based method and ligand-based method. Here structure resembles to the structure of biomolecule, like protein, DNA, or RNA. So if you have information about your structure, then we can proceed for structure-based method, which could include molecular docking or MD simulation and others. But in case if you don't have information about the structure of your biological target, which is biomolecule, biological molecule, we can proceed for ligand-based method if we have information about the active ligand. So for example, imagine you don't know the biological target of drug molecule called paracetamol. So in that case, like for example, you have in the whole world you only know about the paracetamol molecule is there and it can reduce the fever. That's the only information available with you, and you want to design the molecule or something which is better than paracetamol. In that case, we will proceed with ligand-based method because we don't know about the structure or anything else about this particular drug molecule. So we will go with some other methods which are mostly included in ligand-based methods like fingerprint searching, 2D QSAR, 3D QSARs, pharmacophore, or shape-based screening. So these are the various methods which are broadly classified in computer-aided drug designing methodologies. So I would like to, before proceeding further, I would like to emphasize on something like how to design the project. I think that's very critical for any researcher before proceeding for the actual project, if you know about what is your biological target, how much information is available about it in the literature, in the databases, or whatever is experimental data is available with you. So that are the things which one should know before proceeding for the project. So for example, I could divide it into 4 quadrants, and you can search literature based on your these broadly classified quadrants, like whether you have information about only ligand, or whether if you have information about only protein, or you know about protein and ligand, all information about it, or whether you don't know any information about protein, means you don't have structure of protein, you don't have information about ligand. So there are kind of 4 quadrants. So based on that, you need to decide that yes, this is the information which is available with me, so which method I should proceed with. So for example, if you know only information about ligand and you don't know the structure of protein, or you don't know what is the biological target for this ligand, so you cannot go for structure based drug design in that sense. So you need to limit yourself to ligand based drug designing, pharmacophore QSARs, or core hopping, shape screening, such kind of methods which are only relied on the information of ligand. Similarly, if you know only about protein structure, in that case we can use for example that protein structure, and we can proceed for the virtual screening of the data set of compounds, or you can also generate a e-pharmacophore, or you can go for fragment based drug designing or D-node drug designing. If you know about the information of protein structure as well as the inhibitor compound, like drug molecule or any ligand, we can go for structure based drug designing, like molecular docking or MD simulations. So those are the various things which we need to consider. But what if you don't have information of protein structure as well as the ligand molecule? So if you don't have information of protein structure, then there is a methodology called homology modeling. By using homology modeling, we can generate a protein structure, and then you will shift to the protein quadrant from this no information quadrant to the protein quadrant, and in this way you need to proceed for your further project. So that's the information which we always need to keep in our mind. Like first, the very critical thing is literature survey. So that should be very thorough, and you should know about everything about your target, your ligand, or about your project. So that's the thing which we need to be sure about it. And now I will proceed with the particularly structure-based drug designing, and rest of the methods we will discuss in future presentations. So in this presentation, we will mostly talking about molecular docking. So basically, molecular docking is nothing but to identify, we are trying to identify the binding pose of any ligand molecule inside the binding pocket of protein. So that method is, we can call it as a molecular docking, and these such methods are implemented in various software, and with the help of software, we can basically identify how ligand fit inside the binding pocket. So it will give you the protein ligand complex structure, I would say receptor ligand. So what one can expect from the molecular docking is first of all, like binding mode of the ligand. Then we can use this molecular docking methodology for the heat identification or lead identification or optimization, like lead optimization or heat optimization. Means you can perform various substitutions on a ligand molecule, and then you can basically design a new ligand molecule which can have better binding. Then you can also expect some scores, like a score for each binding pose, and based on that score or based on that confirmation, we can select or prioritize few molecules out of all this exercise. So this is what is the brief video of process called molecular docking, and this is what we are planning to do in consecutive slides. So we will have a protein structure in our hand. We need to prepare your protein structure so that your protein structure should get fully prepared and get ready for the molecular docking. We perform molecular docking of millions of compound or data set inside the protein structure, and then out of many such compounds, we try to identify a single molecule which fit best amongst all those data set compounds. So whatever we find best is, we call it as a hit molecule. And then by using or visualizing this compound and interaction, we try to modify further that molecule, keeping the core constant. We can perform various substitutions. And again, we need to perform molecular docking and see how it interacts. And again, based on the binding free energy or docking score, we can prioritize the perfect molecule out of our complete exercise. So that's what can happen virtually with the help of software. And my idea is what are the things which we need to take care of before proceeding for molecular docking. Means these are the some of must do things, whether to use any other software, whether to use Schrodinger, but these are the things which everybody needs to do it. So for example, we can, so this is the general protocol which we need to follow for molecular docking. So that starts with your protein structure. So once you have protein structure in hand, you need to first of all prepare your protein structure. So preparing protein structure is nothing but means you need to, whatever protein structure you get it from crystal structure or NMR data, you need to assign proper bond orders. You need to add hydrogen in each atom. You need to, if it is a metalloprotein, then you need to take care of metal bonds, coordination bonds. If there are some disulfide bridges which are necessary to stabilize the protein structure, that also you need to take care of. You need to convert some modified residues into natural residues in case you critically need to add if there are missing atoms, missing residues or missing loops that you need to add. You need to take care of waters or ions. You need to properly assign the internal hydrogen bonding network at the particular pH which you are interested in, and you also need to refine it, refine the structure using protein preparation, using energy minimization. So these are the things which you need to do. Then next thing related to protein is you need to identify the binding pocket, whether it can be based on the co-crystallized ligand present in it, or it can be based on the druggability of various pockets which are present in the protein. Like in protein, there can be 10 pockets, but out of 10, there will be only single pocket, which is your ligand binding pocket. So you need to identify it. So there are various ways through which one can identify. One of the way is site map. Site means mapping of various sites. So that tool is also present in it. Then regarding ligands, you need to have your ligand in hand. That you can download it from various databases, for example, Zinc or PubChem or DrugBank, and there are many such databases available. Or you can draw your own structures, or you can enumerate the library based on your scaffold or based on your substitutions which are of your interest. For example, if you belongs to a chemistry lab and the characteristics of your chemistry lab is you are working on only one scaffold with different substitutions. So that also you can generate your data set of ligand using library enumeration tool, and you can use it for further screening. But before proceeding for the molecular docking, we need to prepare your ligand. So preparation means same way as protein needs preparation, ligands also need preparation. Because of any ligand can have different ionization state, means atom can have different ionization state, different tautomeric forms, different stereoisomers. So that all things need to be considered before proceeding for molecular docking. And then you need to set your docking parameters and run the docking and analyze your result. So that's the protocol in general you need to follow with molecular docking. So now onwards we will see like how glide works and how easy it is to do all those tasks. Though it looks a lot of tasks you need to do, but there are many softwares available which can do these tasks for you, but you might need to switch from one software to other software and there can be various issues. But at least with Schrodinger you can have all things available in just click button option. So we will see now how this glide works. Just in theory, like it is followed by almost all molecular docking software. It's very general, but I will say afterwards I will emphasize on what specific it is with a glide. So in molecular docking, like there are 3 steps. First one is docking, scoring, and the filtering of your ligand. So in docking, mostly it tries to generate various conformations of a ligand molecule inside the binding pocket. For example, you can see here you have a rigid receptor. So normally in molecular docking exercise, your receptor is considered as a rigid body, but your ligand is flexible. So, but there is also option like you can You can make your receptor also flexible, but in general, docking is with rigid receptor, but flexible ligand. So it generates various conformation of a single ligand, and it try to put all those conformation inside the receptor binding pocket, and it can generate various poses of a single molecule in different conformation, and our task is try to see which is better. So next thing comes into picture is scoring. So scoring means based on the ligand dock pose, whatever it is, based on the interactions, it try to assign some score to it, like glide score or docking score or chem score or gold score, which is in gold software. So similarly in Maestro we have docking score or G score. So it assigns some score based on the various energetics, means based on various interactions which are occurring between protein and ligand. So once you get those scores, so you need to see how those scores look like and what is mean by those scores. So those scores are made up for mostly for good enrichment. So the basic idea behind molecular docking is it's a high throughput way of screening your large library. So even if your docking score doesn't correlate with the experimental values like IC50 or binding constant or EC50 values, you don't need to worry about it, because those scores are not meant for giving you better correlation against these experimental values. So what is the main aim of those score is just to identify good molecules and remove your bad molecules. So it just give you that separation, means in scientific term we can call it as a good enrichment. So enrichment means nothing but if you have a data set of 10,000 compounds, so whether your molecular docking software and the scoring function will able to pick the good active molecules from the complete data set of millions of compounds or not. So this is what is meant for. So you don't need to worry about whether it doesn't correlate or it correlates. If it correlates, that's well and good. If it doesn't correlate, no need to worry about it. So only thing which you can achieve is separation of your good compounds from your bad compounds. So once you get those scores, we need to filter it based on either scores or based on your intuition or your analysis. So for example, you get different poses and different scores, and you have, let's say, a criteria of your ligand should make a particular contact with receptor residue. So based on that, you can analyze all those poses and you can decide which is the final pose which satisfies all your criteria responsible for the good binding. So that's what molecular docking is about. But with Schrodinger, the critical thing is the algorithm, which is proprietary and very crucial for any type of molecular docking exercise. So that this algorithm basically follows site point search, means initially it generate various conformation of ligand and then it put those ligand inside the binding pocket at particular point where you have specified your grid box, and then it tries to generate various type of contacts amongst between protein and ligand, and again through conformational search or energy minimization, and in this way it proceeds with the step-by-step of refinement process of your particular docking pose. So at same time it also generates various conformation, and that's called extensive conformational search around each rotatable bond, so that you should not miss any type of active conformation out of that particular ligand molecule. So it tries to explore it in all possible way, try to generate all possible conformation that systematically means for example, there is a rotatable bond, so you need to rotate your bond through, let's say, 30 degrees each, and then in this way systematically it will rotate all of your bond, try all possible combinations and permutations, and it will give you the final result as an output with the particular score. And again, so this method is very quick and very much reliable, I must say. So there are many literature, you can find it on this glide algorithm, why it is very much useful and why it is very much reliable. And so what are the components of this docking score or glide score. So there are various energetic components like it computes coulombic energy, van der Waals energy, what are the lipophilic contacts, hydrogen bonding interaction, metal contact, what are rotatable bond interactions, and hydrophobic or polar contacts. It also assign various rewards to each contact, and based on that it will give you the final docking score. Means the idea is it will consider all type of possible things which could give impact on your ligand binding. So all those things are getting considered in the docking score. That is the reason why I mean the glide score is pretty reliable in that sense for separation of good compounds from the bad compound. So there is very much peculiar characteristics about glide is three different type of algorithms, and that is called as high throughput virtual screening algorithm, standard precision algorithm, and extra precision. So these are the various precisions. Based on those precisions, actually speed and accuracy changes. So high throughput virtual screening is something which is with a low accuracy, but speed is tremendously high, and you can see like it could dock your ligand in within a second. So in 1 to 2 seconds it will dock the ligand, and in this way basically the overall statistics is you can screen 1.5 millions of compound in just one day on around 35 CPU. So nowadays 35 CPU computer is very much handy, and everybody can get it 35 CPUs computer. So that's not I mean before 10 years it was just unimaginable, but now it is everybody has such computer. Then comes to the standard precision. So in this precision is little bit higher than high throughput virtual screening protocol. So it would take like 10 to 20 seconds for each ligand to dock, and in this way you can screen basically 150,000 compounds in a day on same computer. So but in case of extra precision docking, it takes time like 2 to 3 minutes for each ligand, and in this way you can basically screen few thousands of compound. So based on this accuracy and speed, one need to decide which method I should go further with. We will see those methods. At this time, you just remember like we have three different type of algorithms present with the glide, so that we can use it based on our requirement. So always it is preferable that whenever you have huge library like millions of compound with you, one can go for high throughput virtual screening. If you have some few thousand compounds or a few hundred compounds, we can go for standard precision molecular docking. And if you have some double digit number of compounds like 20, 30, 40 like that, double digit, you can go for extra precision molecular docking. So that's what the overall idea is, and it is very much I would say very much peculiar with the glide docking. You never find such things with other softwares. So now this was all about the docking protocol and how it proceeds in the background. But when we actually want to start docking, we need to start with the protein structure, and there are several databases from where you can get a structure of protein that can be solved through various methods like X-ray, NMR, or cryo-EM methodology. So very well-known database is your protein data bank. I guess everybody knows about it, but there are also another databases like European protein data bank or Japan protein data bank. So you can also use those databases to retrieve your structure of your interest. But there are many things or problems, I would say, which comes with those experimental structures. Not all structures are very well refined, like though they it is solved through experimental techniques, but it doesn't give you the exact resolution of all those atoms or their positions. So mostly in case of, let's say, crystallography, we can have a diffraction pattern or electron density map, and through which a crystallographer solves the structure. It puts various bonds and atoms in this particular electron density, and it will give you the structure, complete structure as an output. But that's your final model which experimentalists give you, and I would say that's a very better starting point for your computational studies, but not foolproof starting point. So what we need to do is there are many such problems and we need to take care of it. So for example, if there is a ligand present in it and you have a density of ligand, I mean electron density of ligand, so that you can predict little bit, but still there is a ambiguity between the hydrogen where it is present, whether it is on nitrogen or on carbon. So that thing you need to take care of. Then there can be various, because you know in crystal structure there are no hydrogen present in it. And so here is the classic example of, let's say, acidic amino acid residue and some other residue, and you can see like there is some hydrogen bonding distance between it, but because you don't know where the hydrogens are placed, whether hydrogens are placed on this particular oxygen or this particular oxygen of the acidic group, like is it a charged particular charged residue or a neutral residue. So that's the thing we need to take care of based on the environment, I would say micro environment. And that's the reason why one need to proceed further. Before proceeding further with crystal structure, you need to refine it. Additionally, there can be many atoms which are missing because a crystallographer could not find the density of few of atoms, and that can be extended to residues, means some residues can be missing or some loops can be missing, means the overall some structure which is missing in the protein. So those are the things which we need to take care of before proceeding for the computer aided drug designing techniques. So in this way there are many such problems. One of such is addition of hydrogen. So you know there are acidic residues like aspartic acid or glutamic acid residues. It can exist in charged form as well as neutral form. So one need to decide like on which particular oxygen the hydrogen is going to be added, as well as whether hydrogen is being added or not. So if you don't add hydrogen, that will be your ionized form, is charged form, and if we add hydrogen on acidic group, it will be the neutral form. The same thing is also applicable to histidines. So just like acidic residue has two states, charged and neutral, aspartic histidine has 6 different states. Because it has a 2 nitrogen and hydrogen can jump from any of can be present on either of nitrogen as well as both nitrogen, and they can have different conformation. So that's these are the few things which one need to take care of before proceeding further for any CAD technique. So one of such example is again internal hydrogen bonding network. Like you can see there are many atoms and residues present in the protein, and each residue is interacting with the surrounding residue through hydrogen bonding or any other type of interaction, and we need to basically optimize that particular internal hydrogen bonding network. You can see here there are many these yellow dotted lines. Those are nothing but the hydrogen bonding which is present between various residues with the surrounding atoms or surrounding residue. So these are the things which you need to take care of. This is regarding protein. So to take care of all those things, we have a very good panel which is called as protein preparation wizard panel. So it looks like this protein preparation wizard. And basically whatever problems which we discuss in last few slides, all those problems are being covered through this panel. And so you can see it can fix everything like protonation state, missing atoms, missing residues, missing loops. It can also take care of water or ions or whatever is present there. You need to see it. It also takes care of internal hydrogen bonding network and performing energy minimization. So this panel gives you almost that flexibility, like by default everything is tick, click on by default, which works for the most of the cases. You just don't need to worry about it until unless you have very specific case, but options are there, like you can customize always these panels and you can run your calculations, means protein preparation wizard thing. So it is I would say it's like just a few clicks away from your crude protein structure to the refined protein structure, and those are just 3 clicks. In just 3 clicks, for example, here is first tab, import, process, review, modify and refine. So these are 3 clicks which you need to run, and that's it. It will take care of everything by default, and it will give you the well refined structure as an output. So that what is very much handy, which comes with you with the Schrodinger suit, and we will see in demo like how it is and how easy it is. So you will, most of the problems of your protein is already covered in just 3 clicks. Then at some point you might be interested in knowing whether your structure is reliable, what is the quality of your structure. So there are various structure evaluation parameters like Ramachandran plot or number of steric clashes or bond length deviations are there. So these are also just 1 click away from you. There is a panel called protein structure quality check or reliability report, protein structure reliability report. So you can just open the panel and click on analyze. For example, this analyze workspace button, and it will give you the complete picture. So not only Ramachandran plot, but it also gives you like which are the deviations, which are the problems present in your structure, and holistically like all those whatever parameters are necessary to check, all those parameters come into just one view. Like wherever you find green, it means it is okay. Wherever you find red means you need to take care of this. So in this particular example, it says red at steric clash, means your protein structure has steric clashes, and you need to get rid of those steric clashes by performing energy minimization. And again, you can just check it whether your structure satisfies all criteria. So that just, I would say, this complete analysis is just 1 click away from you. And next thing comes into picture is identifying the binding pocket for the software. And you can identify your binding pocket like where the co-crystallized ligand or inhibitor present inside the protein structure. It is also just 1 click, like you just need to open the receptor grid generation window, and the options are already ticked, like pick to identify ligand. You just need to click on your ligand, and software will automatically identify, okay, this is the binding pocket for this particular protein. So it will just generate the grid around this box. So normally most of the time with other softwares or few softwares, like you need to remove it manually, and then you need to define your grid box. But with Schrodinger, like it comes handy, software will automatically remove this particular ligand from the position, and you don't need to do anything. You just need to click on ligand and run. That's the very much handy option, I would say. So that's all about like protein structure, how you need to proceed with. Then comes to the ligand molecules. So ligand molecules, as I said, like there are many such databases, for example, binding DB, then PubChem compounds, then ChemSpace, there is drug bank or zinc database, which this could consist of millions of purchasable compound. So just as an example, you can see here, according to this zinc database, they claim like there are 230 millions of purchasable compound available in the database, and those compounds are just ready to dock in 3D format. You can just download those millions of compound and proceed for the molecular docking. So because those are purchasable compound, so at the end you don't need to worry about synthesis and all, you can just purchase it from the respective vendor. Other than that, there are many such natural product databases also that could be of your interest in specifically COVID research. So you can also explore few databases, and those are ready to use database which you can use for your virtual screening exercise. So what are the things which you need to take care of is, for example, just like in protein structure, you need to add hydrogen, you need to take care of bond orders, you need to take care of various ionization states, various tautomers, various stereoisomers. So same thing can be applied also in case of small ligands, and you can see here in ligand preparation, basically the name of panel is lig prep. You just need to provide your structural file which you have downloaded from database. For example, you have 1 million of compound. You just need to provide your file here, and then it will automatically generate like various ionization states for your molecule. For example, here for this molecule, this particular nitrogen has different ionization state. So this is basically on which 3 hydrogen are attached. This is +1 charge state. So in this way, there can be a neutral form, means NH2 form of this nitrogen, or NH3 means the charged form. Similarly, there can be various possibility of tautomers which are present there in this particular molecule. There can be, if there is a stereo center, various stereoisomers can be possibly generated from the molecule, and all those things are being taken care through this panel in just 1 click away. Like you just need to provide your file and click on run. So by default, it take care of everything and it will give you the all type of possible structures out of this particular ligand molecule. So initially you can also apply some filtering criteria, like you don't want to go with millions of compound, but you are only interested in, let's say, a compound with molecular weight less than 460. So you can apply such filters in prior hand so that you can save the time or computational power before proceeding for ligand preparation exercise. So that's the way, like you can smartly deal with the millions of compound. Like millions of compound probably are of not of your interest, because one of the reason is for every protein there is profound binding pocket, and binding pocket is kind of constant. Its volume is also kind of though it fluctuates, but it doesn't fluctuate like too much. But there is a profound binding pocket and its volume, so definite volume. So there is no much space available with it. So probably at some point you are not interested in looking at really big ligand or very small ligand. So that these are the things which you need to look for in prior hand, that which kind of molecules are of my interest, and based on that your information or your knowledge, you can apply these filters. So these are the few smart ways like you can proceed for virtual screening efficiently. So again, this ligand preparation is just one click away. Then you are ready with your protein structure, grid file for your protein, and the ligand molecules which are getting prepared. So then it's a time for molecular docking, and you can see like there is a panel called molecular docking, and it is also just 3 click away. You need to provide your grid file which is in zip format. You need to provide your ligand prepared ligand file, which is again in a MAGZ format or MAE format, or you can use any format like SDF or something else. So just need to provide your input files. You need to choose which precision you want to have. So precision means again this same standard precision, high throughput screen precision or extra precision. You need to decide based on your requirement, and that's it. You just need to click on run. It will perform the docking with huge speed and it will give you the results. So once you get the result, it's a time to analyze your result. And again, to analyze your results means there are two ways to analyze it. One is qualitative way, in which mostly we look for the various interaction between protein and ligand, which kind of interaction, how it is, where it is. And there is another way called quantitative way, in which we mostly look for the scores, means numbers. So again, this qualitative analysis is just like a one click away. We have a particular button. You just need to click on your button and it will give you the this view of particular binding pose, in which ligand will be shown in ball stick view. Your surrounding amino acid residues are shown in lines view, and interactions are being highlighted automatically, whatever interactions are present. So that's again all automatic stuff for which we normally with any other visualizer we need to set up all those things manually. So those are the things like which comes very much handy with you. So if you are very much comfortable with looking at molecule in 3D, that's great. But if you feel like it is messy, you can have 2D interaction viewer. So in 2D view, like you can have 2D structure of ligand and surrounding amino acid residue labels, and also all type of interactions are noted here with some arrows and different colors. So it's an easy way to analyze your molecular docking results for your specific compounds of your interest. So that comes with very much handy, like very much easy, just one button away. Other than that, there is a project table. This table is nothing but it is a form of Excel sheet. Just like in Excel, you have multiple rows and multiple columns. It is also same as that of thing. We call it as a project table. So in project table, you will have all entries from which you get from molecular docking and all docking scores of your interest, and it is already it is sorted based on this docking score, and you just need to look for the docking score which is more negative, better the score is. And this way basically you can decide which molecules I should go with. So there is one obvious question which comes from the user is what is the cutoff of docking score which one need to consider? So the answer is like there is no absolute value for it. It all depends on your reference compound. So for this particular docking exercise, I have performed docking of some 2p33 protein and inhibitor. So basically this entry called 2p33 ligand 2 is the inhibitor bound which was co-crystallized. So I have performed a docking of the list of compound along with the co-crystallized ligand in the 2p33 protein. That's the kinase protein. And after performing docking, I found that this particular ligand, which was co-crystallized and has high binding affinity and activity, experimentally proven activity, has best docking score amongst all. So it has a docking score of, let's say, minus 10.3. And rest of the compounds which were designed in the data set, which are present in the data set, are also around 10, means like minus 9 or minus 8, minus 7. But nobody has a docking score better than the co-crystallized ligand. It says like this ligand has a better binding amongst all those ligands. So you can basically pick those ligands from your data set which can have docking score better than your reference compound, means your co-crystallized ligand. So for example, here I could not select any of the ligand from this list. I will look for some other library and I will check whether I will get a docking score better than minus 10. For example, minus 11 or minus 12. If I get such docking score which is better than the reference compound, I will pick those molecules for my further screening. So that's the thing, means there is no absolute value or cutoff value for the docking scores. It's all relative. You need to think about the reference compound or known drug molecule. So this is where basically we analyze the molecular docking results, and this is the complete protocol for molecular docking result, how to do it. And then based on our previous knowledge, like I have prepared a protocol which one could follow for the docking based virtual screening. So which could start with the, let's say, your protein structure. You get your protein structure, prepare your protein structure through protein preparation wizard. You can generate a grid of it. Then you can see whether your grid is fine, like by visually looking whether it covers the all binding pocket or residues of your interest. So same thing you need to do with ligand. You need to download your ligand molecules from some data set, or you can draw it, or you can generate your own library. Then you can apply various filters on that library, like Lipinski filter or other drug likeness filter or any other filters, molecular filters which is of your interest. Then you need to prepare that library of ligand using ligand preparation module, and you can proceed for the molecular docking. So now comes the flow, like which various type of docking algorithm should I use. So for hit selection, like for example, let's see you are dealing with some zinc database or any other database which consists of 1 million compounds. So if the number is huge, like millions of compound or few lakhs of compound, I would proceed with high throughput virtual screening because it is fast enough. Through this high throughput virtual screening exercise, I would get like whether out of 1 million compound, whether let's say how many compounds are able to fit inside the binding pocket. That's it. So out of 1 million, for example, there is only 1 lakh compound which are able to fit inside the binding pocket. So I will just select some top 10% or top 1% out of it based on simple score. Probably at this stage, I would not look at the molecular interaction or anything else because I'm not really interested for millions of compound for such thing. Then next comes to the standard precision molecular docking, which is more reliable than high throughput virtual screening. Let's say you have few thousands of compound. We will go with the standard precision molecular docking, and probably at this stage I would look for the all type of compounds which are able to dock inside the binding pocket and form the particular interactions which are of your interest, as well as their score should also be good enough, like more than your reference compound. So I would consider like score and interactions for my second cut. And out of it, like I will select, let's say, top 1% or top 10% of compounds, and I will proceed with extra precision molecular docking. Let's say you have some double digit of number of compound, like 80 compound or 90 compound, let's say. And then for that, I will go for extra precision molecular docking, and I will then visually inspect for each and every compound which comes on the top based on the extra precision docking score. and then I will look into compound. I will check which are the good characteristics present in compound for your receptor binding, which are bad things which are present in the compound, and in this way I will visually inspect all compounds and proceed for further. So next step can be your binding free energy calculation using MMGBSA method, because again it's a fast method to compute it for mini structure. So we have a tool called Prime MMGBSA, and using that tool you can pretty fastly compute binding free energy for your dock poses of the ligand, and based on binding free energy, which is your delta G, should be more negative, and your visual inspection, and then you can vote for your compounds, because the number will be few, like let's say 20 or 30 compounds at the end, and based on that you can select like your top list of compound, like 10 compounds or 20 compounds, and whether to proceed for buying of those compounds you can decide, or you want to go for further molecular modeling exercise, for example molecular dynamic simulation. So one can proceed for molecular dynamic simulation for selected compound, let's say 10 or 20, and out of those 20 you can select, let's say, top 5 compounds from your list, and you can buy those and check it for the actual experiments. So how good glide docking is in picking right kind of compound is this is the study which was published in 2012 in Journal of Computer Aided Drug Designing by various research group based on their experiments. So they found that in case of, you can see this orange color is for glide docking, and they compare glide, gold and dock software, and out of which whenever they computed enrichment of it, you can see in top 0.1%, like I mean as compared to other software, most of the ligands were active ligands were picked. If you see top 1%, around 25% of active ligands were picked, and in this way like 100% in all of your dock pose, like most of your active compounds, like 80% of active compounds are being picked for using this glide method. So that's why initially I said like glide is pretty reliable in that sense for picking active compounds. So as such you don't need to believe on me whatever I'm saying, but there is a lot of literature which is available there. You can simply search it and be sure about it. So coming towards the end of presentation, that's the whatever we have seen was the in general how to perform molecular docking, how to analyze it, but that's not the complete story of molecular docking. But critical parts come afterwards, like there are many types of docking. So far we have just seen high throughput virtual screening, standard precision or extra precision docking, but there can be a case when you are dealing with the peptide compounds or peptidomimetic type of compound. So for that it requires a different treatment, because peptides are more flexible than your small organic compounds. If there are metallo compounds, you need to have different treatment. So if there are macrocyclic compounds or natural product, it needs different treatment. If there are covalent binder, that's the case with COVID, there are covalent binders. So that needs different treatment. This QM polarized means quantum mechanically polarized ligand docking. It is mostly, we can use it for the metal containing ligands or proteins. So that is also take care of. There is a method called induced fit docking, in which basically we apply flexibility to the protein target. And there is also a term called cross docking, means we can dock inhibitor of one protein into another protein, means another isoform, and vice versa. In this way, that methodology is called cross docking. So all those things are getting covered in the glide docking, which missed by most of the software. But again, like this is what the complete story is. The take-home message is every type of compound requires a different treatment. Different compounds, different treatment. One should not consider a single treatment for all type of compounds. There is nothing such thing called robustness in, at least in docking. But most of the people they lack there, and they consider like a robust treatment for all type of compounds. But you need to always keep in mind that compounds are different, treatment should be different. So that's what the take home message is. We have all that options present with us. We just need to explore those things. So here onwards, we are going to see the Maestro interface, how it looks like and how actually one need to perform molecular docking or protein preparation or ligand preparation and all handling things. So that will be taken care of my colleague Vinod. But before proceeding to this, probably just you need to spend 1 minute to see this is the Maestro interface. There are some critical things which are present in all type of softwares is their interface, and you need to know which are the options which you can see in just one go. So those options is whatever this region which is present in a black color background is called as your workspace. Here you visualize your molecule, you can see it, you can change the color of background, that's not a big problem. We call it as a workspace. Then on left hand side you will see the table. That table is called as workspace navigator, in which basically there are various entries. Whatever you do, whatever you import, those all comes in the form of entries, and finally it comes in the form of table. There is a section called on left hand side bottom structure hierarchy. Structure hierarchy is nothing but whatever structure is loaded on workspace, how many components are present in that workspace. For example, there is protein, a single component, a ligand, other component, metal ion, other component, water, other component. So these are the various components you can see in structure hierarchy. There is an option for quick select on the top left. So you can select an atom, you can select a residue, you can select a chain, you can select a molecule. Those all options are present. There are some quick selects, like quickly just by pressing P, you can select protein, L for ligand, S for solvent. So that's I would say pretty user friendly. You can select it. You can change the representation using this style box. There are in style there are various options. You can change the representation to lines, tubes, ball sticks, space fill. You can change the color. You can change the cartoon representation or ribbon representation or surface view. So there are many such options which are present in style. So you need to remember, yes, whenever you want to change the style, you just need to look for this button. Then there is a, you can see 2D overlay button. Basically it shows which ligand is present in it, and it says the 2D image of this ligand. Then there are few toggle buttons. I would say that's more important. So toggle button is nothing but related to the representation, like you want to see cartoons view, you want to see surface view, you want to see interactions, and we will explore more options in actual demo. And you can also have in bottom status bar where you can you will know like how many number of atoms present on the workspace, how many number of residues present on the workspace, what is selected, what is not selected, all those things. The most important option comes with the task bar, task tool. So basically this has all options, means it has access to all modules like protein preparation, ligand preparation, docking, grid generation, pharmacophore, MD simulation. All modules are present in this task button. So in next thing we will try to explore all those options, and the session will be taken by my colleague Vinod. So here onwards I will hand over to Vinod.
Yeah. So can you just stop sharing? I'll try to share then.
Yeah.
So can you see my screen?
Yes.
Am I audible? Loud and clear?
Yes.
Okay. Thank you, Prajwal, for such a fabulous introduction. So my demo will purely focus on how to use this software from a no voice angle. My talk will try to cover. I hope there are some echoes, Prajwal. I hope you are in mute.
Yes.
Okay. So the first thing that see the interface, this is the interface, how it looks like. Maestro interface is pretty handy and it has a single click orientation. The thing that you need to focus firsthand is I've seen many students doing this mistake. So I request everyone, please change your directory and work on it. So by default, it comes in a Schrodinger directory, and but I wish to give a demo. So I have a folder called as test docking, and I'll choose this. And once you have done this, all the files will obviously go to that directories. All the softwares are available in task as rightly pointed out by Prajwal. It is straightforward. The tools are available. In case you are not aware how to search, you can just type like in Google. Like for example, if I do docking, you can see the various kinds of docking Schrodinger provides. If that is done, as I said, my discussions were purely from a project point of view. So anybody who I think I have seen from no voice angle, that they will go into PDB and select their PDB of choice. For example, I'll show the demo on this J&K inhibitors. As a chemist, as a drug hunter, what you need to focus is first on the bound ligand. What is the activity, biological activity it has? If it is in nanomolar or picomolar, then your chances of identifying good molecules would be obvious. But many of them mistake by selecting PDB reported earlier. So I request everyone, please evaluate that properly and go ahead. So I will try to download this PDB and explore what are the other options we have. So first you need to click on file and say get PDB. And here the option is straightforward. You will just give the PDB ID and I'll try to download it. Once the protein is downloaded, this is how the view looks as pointed out by Prajwal. If you see the protein, it's always handy to see the sequence details. That can be accessed from this window panel. If you click on window, you have the sequence viewer details and it comes like this. So you can drag this up and you have the secondary structure details as well, which can be switched off and switched on as per choice. Now, another very, very important thing which many researchers miss is whether there are any breaks. As Prajwal was pointing out in the presentation, there are possibilities of having a break. So this you can see that a single click, you can identify there are breaks available. So this is one point. The next thing that you can focus, if you click L in the workspace in the keyboard, then you can see the protein. So how am I'm doing the control, how to control the structure in the workspace. So what you need to do is you can use shortcuts like dot, semicolon, question mark and comma, and J and K will help you to zoom in and zoom out. So if you are using mouse, then right click can help you to move the structure up, down. Center click will help you to move the structure in 3D. And then the selection is your left click. So if I select anything, automatically it get highlighted in the sequence viewer. There is a nice handy shortcut, Z or Z. If you click that, it will focus. So if you quickly want to get out of this, there is an option for deselection, or else you can click in an empty space to get it deselected. If you click Z, you are back in space. So I've shown you the secondary structure in the sequence viewer. Okay, what if I want to see the secondary structure in the protein? So here is your ribbons. So looks interesting. Yes, there are many ribbon views, but what you need to focus is the ligand, right? As a drug hunter, you need to focus on what are the critical interactions the ligand has. So I'm purely talking with respect to computational chemistry. So don't mistake me. I should not miss the critical points. Here I have my protein and you can see the structure hierarchy as subtasks. So I want to apply the protein surface so that I have better view. So this is the view I get where I can clearly see in the surface view how the ligand sits. And also it is tricky to see, okay, how is the spread of the solvent, which solvent to keep and which to not. So this is the view you get. And clearly, can you see those who are new to molecular modeling, please note that the red color is oxygen, the blue is nitrogen. Okay. Other issues are like you could notice this interaction doesn't show any interactions, as Prajwal was pointing out, right, in the analysis, because it is in raw form. So we have a single click solution for all these problems. The first part is the protein preparation wizard, which can be accessed here, or else you can go to task and type the protein preparation. So if I click protein preparation, so this tool, please keep the panels in the right side, whatever the software you may use. The first thing that you need to do, just for clarity, I'm removing this surface. And all the options is already explained by Prajwal, so my work is quietly reduced. I'll just hit pre-process. But please note that there is a fill-in missing side chains using prime and loops. So I have already filled it and added it for you. So let me just import that structure for you. So all the structures are automatically saved and the loops have been filled. So this is how the structure looks. So now you can see the loops have been filled up. So it has been taken care. So just imagine if it is in the binding site, and in case if you want to perform molecular dynamics, then it becomes very, very critical. So virtually I have done this part, that is pre-process. I'll just do it again. So I'm bypassing that fill in missing side chains and loops due to the fact that I just want to save time. So this part is done. You could see there are overlapping atoms, which is the issue, as these two atoms are overlapping, or this one is overlapping, or this one is overlapping, and so on. If I proceed further, there are many components, like you have a single chain and you have various water molecules which need to be taken care. And there is an inhibitor available that also has to be taken care. And here we have, but see, water molecule, we will have a separate debate. I'll just show you. In case if you have multiple chains like A, B, C, D, try to retain the chain that has the inhibitor. If the inhibitor is in the binding interface of both chains, like in COVID situation, so please you need to retain both the chain. Here we have refine. Refine options helps you to take care of the series clashes, which I said. And if I hit optimize, it will take care of the optimization. The job is running in the background. All the jobs are shown here. You can see the status of the job. If you want further details, if you hit here and say monitor, you can see the running job. Our tech support always helps this, like, because it's not always a smooth affair that your docking is going very smoothly. It's not the case. So when things are running and if something goes wrong, we have a nice option of postmortem, which you need to send it to help@Schrodinger.com. So if you do that, they will get back to you explaining what went wrong. For the best learning in computational chemistry, more errors, more issues will help you and create a foundation so that your outcomes are always best. So that part, because it is running and I want to save time, I already prepared this. Okay, see here, there is an option for remove waters. So what I will do, I'll just make this as 2. Okay, so how to retain or what waters to retain. So if you mention here 2, and if I hit remove waters, then all those waters which forms a bridged hydrogen bonding will be retained and rest will be removed. So the water molecule that forms a bridged hydrogen bonding are the critical ones. So I'll just show you what they are exactly. If I hit here in the review and modify, and if I say this water molecule, this is 603, see currently it is not showing any hydrogen bonds, right? And the best, see, as the toggle buttons are available, here is an option for activating the non-bonding interactions. If you do right click, you can see all the non-bonding critical interactions like pi-pi interaction, halogen bonds, hydrogen bonds, salt bridges, and various contact clashes and all that. As I said, that's not my point of discussion. Here it is not making sense. Okay, yes, it's making 2 hydrogen bonds, but with what? I'm not able to clearly understand. We have a single click option, which call it as preset. Double click this, and you can see an excellent view where things will become much clearer. As I said, bridged hydrogen bonding. Bridged hydrogen bonding is water molecule that bridges the ligand and protein. So this could be critical water molecule that if deleted, then all your calculation may end up in a mess. So that's done. Then the final thing to take care is minimization, which I shall do. If I hit minimize, then it will try to minimize the complete system quickly for you using the force field OPLS3E. Please, you don't have to panic because it's an X-ray structure. During minimization, it will converge if your RMSD changes from 0.30 Armstrong. I think that will retain the experimental integrity. So if the job is running, you can see. So I just showed you how to select a protein. Then now I am showing you how to prepare the protein, and I'm in the last stage. So then once done, how do you validate your protein? If I'm in your place, what I will focus is purely first on the ligand interaction, whether all the crystal interactions are reported properly or not. You can see it has an interaction with 149, 152, and a water bridge interaction with lysine 93. So that's what is focus. So now what you need to do is the next thing, because it is related, protein related task, and you can see it is really messy, right? You can see a lot of labels, so that can be, you can hide it here. So if you click this label, it will be removed. You can remove the ribbons too and activate it. You can activate the non-bonding interactions and so on, because purely the focus is on docking. So I'll try to just preset this and focus on the protein ligand complex alone. The next task is to identify the binding site. There are specific tool called a site map in case you are not aware of your binding site. So this is a nice tool, but right now I'll not explain much, but just keep this in mind. Luckily, in this target of choice, we have the ligand in it. So once for that to specify the grid, what you need to do is you need to click glide receptor grid generation. So as I Prajwal explained, it is a single click solution. If you click this, then your grid is pretty good to go. So there are more advanced options which will help you later, but please focus on the critical things. So I'll give a job prefix of 2p33 and I will hit run. So the grid generation is running. So that is all what I have with the protein. Let me move to the ligand, which is a very critical part. So there are various ways of bringing the ligands. You can click file and import structure, but currently I will show a very basic 2D sketching. That is phenol. Phenol is my favorite structure because I'll not end up in any errors. Here you go, and I will just save this. So this is S2, and I think it's same like your ChemDraw and ChemSketch, so should not be worried. If you hit Z, you can see the structure. This is what I see. In case if you want to sketch in 3D, the options are available in build. If you click build, then you have add fragment option where you can sketch whatever fragment you have. We have metal center options. We have various diverse fragments that you have. OK, so whatever you want, you can select it and place it. That looks fine. OK, so you have a structure. That's good. But what if you want to do, because single structure sketching is quite tedious, and Prajwal already explained there are online database that you can use, but many classical chemistry legends will try to exploit their own course for anti-COVID situation. So in that case, you can enumerate your library and increase the capability of your molecules. So how you can do that? You just see the cursor is with the letter A. In case there are various options for selections as how you can pick your selections. Currently, I will do atoms. So I have just selected this one atom and here one more atom. I will try to do the enumeration. The enumeration is pretty simple. As I said, in build, the options are there. Create enumerated entries. In create and enumerated entries, see right now, the combination is going to end up in a very huge number of structures. Due to limitation of time, so I won't do that now. I will try to select a very diverse R groups and solubilizing R groups. That should be fine. So I'll give a suffix 1FJS and hit run. So it's not going to take time as it's going to generate only 1419 structures. But for our docking, we have structures already in hand. What I will do, I will import a set of already available SMILE structure. So if you notice here, I have my structures placed inside my working directory. That is, here is the ligands and here is the input ligands. So these are all SMILE structures. You can see it's all in 2D format. So I have 5 structure in place, and these 5 structures I need to prepare it as pointed out by Prajwal in LigPrep. The LigPrep tool will help you quickly prepare the structures. But I have 2 aspects of it. Please, you need to focus that the databases, if you are dealing with bigger databases, you need to carefully select the options. If you're going to select by default, it is going to increase the size of your database. I think luckily my database, that is the enumeration also got over. I'll show you that result also. And here in LigPrep, I can say project table selected entries for only this 5 because of limitation of time. So project table selected entries, and by default, you can say hit run. 1F.js is ready to go. And please note down, you can increase the capability of your speed by increasing the number of processor. So I can just try to use 5 subjobs with my 5 processor so that the molecules get distributed and I get quickly the results. So, but in case if you're dealing with database, I request you to keep it in neutralized unless you are aware of your pH. Otherwise, you can but keep this desalt on, remove the generate automers, and don't generate combinations of your stereoisomer center also, because it will end up in increasing the size of the molecules. Just by default, I will be preparing the structure that will give me various variants of ionizations, stereoisomers, and tautomers too, as you will see the results. Okay, let me just show you the enumeration results. You can clearly see here the outputs are available. All things can be easily organized by this entry list. The entry list also has option to split. For example, in this case, I can split the protein ligand, and this is the inclusion and this is selection. Okay, and here you have the enumeration library. Let me just show you. The enumeration, as I pointed out, it's something 1,400 odd structures. I'll just show you in a 2D view. So here is my collection of structure. There is another interesting option also, which will be liked by various synthetic chemists. Because it's the urges for molecules. It's not to end up just with papers, not synthesizing the molecules. You need to have rationality in generating the libraries. We have an option called as reaction-based enumeration, which will help you to identify your reaction of your molecule. Here you go. Here is a structure, and I will hit generate pathway. I will get various combinations of reaction that can end up with my product. So, and in case if you feel no, I'm not able to find the reaction of my choice, all the reactions are listed here. You can also sketch and create your own reactions too. That option comes handy if you have a novel reactions. I know many chemistry has their reactions novel, so it's not, it maybe not be available in the existing reaction-based enumeration. So you need to add it. It's a very basic add-on, and that's how you can generate the various library of molecules. So as I already covered many things, so this is the critical part, that how to go about docking. Docking is pretty straightforward. We'll go to browse. Here is a glide. You can give as a shortcut, as I earlier mentioned, and here is glide, and you have ligand docking. Ligand docking is like, actually, see, according to me, for doing docking, you need to know 3 things: where to dock, what to dock, and how to dock. So where to dock is the grid, the protein grid that we generated during grid generation. And what to dock is the ligand molecule that I prepared during ligand preparation. Please note that you have multiple options of selecting it from project table also. This is a small collection of molecules. And how to dock, that is standard precision, extra precision, and high throughput virtual screening, as pointed out by Prajwal. And in the output options, you can mention how many poses you generate and what are the per residue interaction among 12 Armstrong grid center and so on. I will try to focus on the output. I'm not running it, please. As I said, due to time, we need to run around. So here it is, file, import structures, and in my working directory, I already have the poses placed. Here I have the SP docking results. If I open it, you can easily see the collection of molecules in the entry list. So the first entry is always the protein followed by the various ligands. For our comfort, we also have the ligand placed, the native ligand. So how do you analyze? I usually start with 2D interactions. I will try to include all the structures at one click. Here you go. So all the structures have been placed. You can see all the docking is been, all the poses of the molecules are replaced here and their interactions. So for the comfort, I will remove the contacts now that are bad and ugly, but that also plays critical role in selection and rejection of molecules. The molecule that has least bad contacts can be ignored. In this case, when I go and hit the ligand interaction diagram, you will notice this is the native ligand, and you can qualitatively analyze, okay, what are the kind of interaction it makes? Which is that molecule that gives me the similar kind of interactions? This is one way of selection. The second way of selection would be to go with docking score. There are many properties available, but to be precise, this is the property tree. The property tree can be particularly selected for only glide-based properties. So I have my glide and I will activate only glide primary properties. So these are the properties I have. It is pretty easy to export this in form of Excel, and also it is pretty easy to showcase as a 2D plot. So this is the 2D view. This is the 2D view, and you also have a 2D viewer, which I earlier showed you. Now you can also analyze in another aspect by pose variations. That is, I think if I generate the pose, means just by orientation, no scoring, it is purely intuition based. I'll try to add the surface for the protein. And now please all of you watch carefully that I have fixed this protein, and I'll try to play around and see which are the best proteins that are highly complement to the target. The options are pretty straightforward. So let me just play this. So hope it is visible. So your intuition will help you to understand and decide on which ligand to select. And okay, yeah, in case we know that looks fine, but can I get an overall summary of this? Yes, Schrodinger has an excellent option for analysis in that way. We have a tool called as SIFT. Okay, and if you say generate fingerprints, at one go, it will go show me all the interactions of all the molecules which I generated in a single plot. I think that's all from me. Prajwal, really thank you for the opportunity, Dr. Giri and others coordinators of Drug Discovery Hackathon. Let's see if we can meet on one another time. Thank you, Prajwal. It's your turn. Prajwal, are you there?
Yes, yes, I am there. So Vinod, could you please put on mute? Yep. Okay, great. Yep. So just a slideshow, and so I hope you are now pretty much familiar with the Maestro interface and how to perform actual molecular docking analysis and library enumeration. And I would say, I mean, it's pretty easy if you give a try. And it is very nicely explained by Vinod in very easy way. But always, like person to person, the opinion can change. But you can always look into more detail, or you can always look forward to use such software. So other than that, as I always mention about the help button, like you always have help button with you, which gives you all possible type of help which is related to the Schrodinger software. And we are also there to help you out with all type of possible queries. So don't forget to contact us. Don't forget to try the help option once. Everything is there. You can simply search it just like a Google search. You will find all things possible. Then other than that, we have various internal resources which are also available for you, like knowledge base, in which it answers most of the queries raised by various users. So you can find various type of discussions, for example, what is docking, some basic discussion, or what is difference between docking scores and such kind of discussions. You can always have those discussions in knowledge base. The link is here, as well as I mean, you can have access to all. So don't worry about it. Just to go to Google and search for it. You will find this thing. Then we have a support center, training center, which consists of many tutorial videos for various modules that also you might want to explore. We have various seminars, and those all are from various Schrodinger scientists. So the easiest thing is you just need to go to schrodinger.com. And first thing is get registered yourself. You just need to put your name, email ID. That's the minimum information, like you belongs to academics or industry. Such things you just need to provide. And once you get yourself registered, you will have access to many such interesting materials. Other than that, as I always said, like we are always there to help you out. So at the end, I would like to acknowledge my team. So we are the team of 8 who are located in Bangalore office. So we are all of us are ready to help you, and you can always contact us anytime whenever you have any questions. So we are also dealing with the material science, so you can also contact us for material science questions. So with this, I would again like to thanks to the organizers of Drug Discovery Hackathon 2020 and Dr. Shastri and the coordinator Dr. Giri for giving us an opportunity to talk, to give them overview of glide docking protocol and analysis. So again, I thank you, all of you, for joining us for a session and listen everything carefully, I hope. And I would like to open the session for questions if you have. And again, like I would like to mention that our email IDs are here. You can contact us. Don't hesitate to contact us. And I will hand over to Dr. Giri if we have some questions. I would be happy to answer those questions.
Absolutely, Prajwal. Thank you so much for this wonderful session, both to Prajwal as well as Vinod. As you rightly said, we need to come away from black boxing, and we need to understand each steps and move forward. So there are plenty of questions. Due to time limitations, we will pick up few of them, and then probably any one of you could answer. So here it goes. First question, is there any machine learning methodology or pipeline does Schrodinger have for target identification based library screening?
It's so basically machine learning is like still it is developing in the field of drug discovery, and we also have a machine learning modules which are also based on deep learning method. So, but that is for not for docking, but it is for QSAR studies. So we have a module called Deep Chem and Auto QSAR. through which one can use those machine learning or deep learning methodologies. But for docking, I think it is mostly physics based. So that's the reason why you still don't have docking through machine learning.
Yeah.
Because it's a physics-based research.
Okay. Another question, whether tautomers and stereoisomers are considered as different structure during docking in your software?
Yes. So not during docking, but while preparing your ligand itself, it generates various tautomers and stereoisomers. And ideally, you must use those structures for docking. And at the end of docking results, you can see which stereoisomer is better, which tautomer is better. So ideally, you need to explore all possible structures of a single ligand.
Okay. Another question, is there any protocol or workflow available that you can do docking with one ligand on multiple receptors?
Yes, that's a great question, I must say. And seems you are very keen to your research. So yes, such protocol is available with us. It's the same like a molecular docking, but you just need to perform it repeatedly for single ligand in different protein structures. But because again, it's a multiple task, you need to do it repeatedly. The same task you need to do repeatedly. So that's the reason why we have created a protocol, a panel called cross-docking or virtual screening workflow. You can use it to dock a single ligand in multiple protein structures at a time. You don't need to do it repeatedly. It's software will take care of everything.
Okay. Another question is for Zinc database or other similar databases, whether one should process the ligand, like adding hydrogens or 3D optimization, generating conformers or anything like that before docking.
Okay, so ideally, yes. But if you say whether I need to add hydrogen, so that depends what you are going to download. So just as an example, even from Zinc database, you can download it in a 2D format, you can download it in a Smiles format, you can download it in a 3D format. So if you are downloading it in a 3D format, then it means the complete structure is present, hydrogens are there. But still you need to go for ligand preparation because you need to explore possibility of tautomers, ionization states and stereoisomers. And that information is missing in those structure. So you need to still go for ligand preparation.
Okay.
So
another question.
Add to it, just as I said, sampling the conformation before docking will be tricky because global minima and bioactive minima are totally different. So though you generate your global minima conformations, at the end of the day, the docking program will try to generate its own conformations. So it is not mandatory to generate conformers.
Absolutely. Yeah. Okay.
Yep.
Another question is that how can we perform ensemble docking? And do you have any benchmark on how much time it could take if we have like 5 to 10 crystal structures?
So again, it's depend on number of ligands. So for example, yes, you can do first of all ensemble docking. Ensemble docking is nothing but the multiple protein structures with you. Just a single protein structure has multiple conformations. So that leads to the multiple protein input structures. So yes, you can do it. And then it depends on how many ligands you want to dock. Let's say one ligand. So you could consider the time of one ligand would require in standard precision, it could take like 10 to 20 seconds in each protein. In this way, you can consider the time.
Okay. Another one general question, but I hope it's a right platform because Drug Discovery Hackathon is open to everybody. So one question is here is that what are the criteria that one needs to consider while selecting a protein structure from the PDB database or even for docking?
Okay. So that's the huge topic of discussion. But again, there are some straightforward ways to select it. Just like Vinod explained, first of all, you need to look for whether crystal structure is available for it or NMR structure is available for it. Then next criteria can be whether there are missing atoms, residues or loop. then what is the resolution of this particular crystal structure. Then you can also look for the co-crystallized ligand, which co-crystallized ligand is present in it, what is its biological activity value. So one should consider like a most active ligand if you are proceeding for the drug discovery or drug designing. So you can take a most active ligand inside the binding pocket, which is co-crystallized, and which it should have high resolution possible. It should have less missing atoms residues possible. And again, you can go for a crystal structure. Again, like you will have like NMR structure, crystal structure, and cryo-EM structure. But at some point, I would prefer crystal structure because NMR structure might have different conformation of residues, and cryo-EM has not that much great resolution. So that's why high resolution crystal structure. So these are the few in general criteria.
Okay. Another question. Many people have asked this question. Can we or how to perform, or is it possible to perform docking on organometallic compounds?
Yes.
Well as natural compounds, extracted natural compounds.
Yes. So as I said, like glide is kind of a complete solution, and you will have different treatment for each different type of ligands. So yes, organometallic can be performed. So you can use either molecular mechanics or force field based charge derivations which are present in the simple glide. Or if you want more precise results, you can use quantum polarized molecular docking, in which your metal ion and your ligand and interactions will be treated at quantum mechanics level. So I would always prefer to go for quantum polarized molecular docking for organometallic compounds.
Okay.
Just to add to that, Prajwal, actually organometallic compounds are quite tricky also. A lot of research is still in progress because metal coordination based force fields are very tricky. But Glide has various different publications. So it is always advised that you optimize your organometallic compounds using quantum mechanics, then followed by rigid docking or QPLD. It should help you.
Yeah.
Okay. And the final two more questions. There are many more. Due to time constraint, I have to limit to two more. One is, what is the procedure to add missing amino acids in the proteins? If it is like there are missing amino acids in the protein using your software?
So I think that point is already covered by Vinod in the session, like in his particular test protein. There were missing residues, and in protein preparation wizard panel itself, we have a option which you need to just tick, and it will add all those missing residues in it, missing atoms, missing loops. It will be taken care of.
So if you're asking the background algorithm, that actually in all the PDB, the FASTA sequence details are available. So our module prime actually adds it by default. By knowledge-based or by energy-based add-ons.
Okay. Okay. Now, based on your answer, another person is asking, how do we define the binding site in case of one ligand with many receptor docking?
One ligand with many receptor docking. Yeah. So what is meant by that actually?
Earlier there was a question on one ligand with multiple proteins.
Yeah.
So on that context,
there are two kinds actually. So our cross-docking option has multiple options. Like one is predefining, if the native ligand is there, that will be automatically picked as a grid centroid. And in case if that is not there, there are options for generating it using sitemap. But doing too much automation will mess up with our work.
Absolutely, you're right.
So you need to be careful actually, because drug discovery based things are quite minute and need to be taken care very carefully.
Absolutely. That's where we come to know that a good synergetic between human and computational expertise will show wonders. It's not only computational efforts. Great. Thank you so much, Prajwal and Vinod, for this wonderful session. There are very great comments from the chat, and I thank both of you. And I also want to thank the people behind this to organize this from NIST, especially Professor Narahari Shastri and his group members listed here in this particular slide. I wanted to also repeat again, these are the official websites, the registration link for hackathon as well as for certificates, everything will be announced very soon. So kindly be patient, and training sessions will be always in this channel of MHRD's Innovation Cell. But as I can see, many of you are asking the questions of do's and don'ts in a drug discovery hackathon. Please listen to Abhay Jerry's a video which is in his channel where he has talked about how the team formation, who could be in the team, what are the do's and what all things you get access. Now, one more question that came up in the chat was, how could we get access to all these softwares provided by many partners in Drug Discovery Hackathon? So please wait until your official registrations gets opened. Once you're registered, you will have all the details through the Drug Discovery Hackathon virtual tool room from CDAC. So there you will have the documentation to help you how to get access to all these softwares. So with that said, thank you all. Have a very good evening and bye-bye.
Yep. Thank you, Dr. Giri. Thank you very much.